human insulin. It was launched by an American company named Eli Lilly in 1983. Human insulin is being commercially produced from a transgenic bacterium, Agrobacterium species. Insulin is the first hormone artificially produced by culturing bacteria. Students, insulin is a protein hormone. This hormone is produced by the islets of Langerhans in human beings. Now, if insulin is not synthesized or insulin is not produced in the required amount, then the control of blood sugar level in the body becomes very difficult, and this leads to what is called diabetes. So, here initially when a person suffered from diabetes, he was given insulin from the pancreas cow or pig, that is from the slaughterhouse. They used to collect the pancreas of cow and pig, and from them they used to extract insulin and give it to the diabetic patient. But in this case what happens, the body of human beings started to reject it, that is immune rejection was there, and some side effects were also there because the amino acid sequence in those animals, that is in cow and pig, were different from the insulin present in human body. So, it became mandatory to start synthesizing human insulin. So, a company, an American company called Eli Lilly, this was the one. For the first time they launched human insulin by the name Humulin. So, this particular thing is developed in such a manner that it will have all the nucleotides of a normal insulin. So, it will not show any immune rejection or side effects. How is it actually produced? Basically, if you see an insulin, it has got 3 chains, 3 polypeptide chains, A, B, C we can see. But for an active insulin, only chain A and chain B is required, C is not required. So, these days how insulin is synthesized artificially by genetic engineering is, the genes required or which encodes for chain alpha and beta, that is the first 2 chains which are needed to produce an active insulin, these 2 genes are incorporated into the plasmids of different bacteria. That is recombinant DNA is synthesized. Plasmid alli gene A anna ಹಾಕ್ತಾರೆ, ಇನ್ನೊಂದು ಪ್ಲಾಸ್ಮಿಡ್ ಅಲ್ಲಿ ಜೀನ್ ಬಿ ಅನ್ನು ಇನ್ಸರ್ಟ್ ಮಾಡಿಬಿಟ್ಟು ಇದನ್ನು ಡಿಫ್ರೆಂಟ್ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಇನ್ಸರ್ಟ್ ಮಾಡ್ತಾರೆ ಹೋಸ್ಟ್ ಅಲ್ಲಿ ಫಾರ್ ಕ್ಲೋನಿಂಗ್. So, a culture is maintained where gene A is present, another culture of bacteria is maintained where the culture the bacteria consists of gene B. As a result, gene A, the bacteria starts producing the alpha chain, and in the next culture it starts producing beta chain. Later A chain and B chain ಅಥವಾ alpha and beta chain ಅನ್ನು ಕಲೆಕ್ಟ್ ಮಾಡಿಬಿಟ್ಟು ಅದನ್ನು ಕಂಬೈನ್ ಮಾಡ್ತಾರೆ by formation of disulfide bond. So, once disulfide bond is formed between these 2 chains, it becomes an active insulin. So, then it is given to the diabetic patients to cure what is called diabetes. So, in the options if you see, Humulin is the genetically engineered human insulin is correct. It was launched by an American company named Eli Lilly is also correct. Human insulin is being commercially produced from a transgenic bacterium is true, but the name of the bacterium is given wrong over there. The bacteria employed is E. coli, not Agrobacterium. So, that option is wrong, and the last option, insulin is the first hormone artificially produced by culturing bacteria, that is also a right option. So, in this question 4, the right answer is option C, because that is a wrong statement. Coded by which of the following genes controls the corn borer? Now, this is an example for transgenic plants. We are aware that there are number of transgenic plants, that is the plants which have some foreign gene incorporated into its body so that it can fight against some or the other pests. So, here by introducing such genes into the host plant, the host plant becomes resistant against some pest or probably some insects. This is beneficial because we need not use any insecticides or pesticides to control these insects or pests. So, it is environmental friendly. Now, from where are these genes extracted? So, BT stands for Bacillus thuringiensis. This Bacillus thuringiensis is a bacteria. It comprises of or it consists of a gene called CRY gene. This CRY gene stands for CRY, stands for crystal protein. Okay. So, bacteria consist of a gene called CRY gene, which encodes for a protein called CRY protein, or it is nothing but a toxin. It is a crystal toxin. That is why we write CRY. Now, there is a slight difference here. When we write capital C, small r and small y, and when it is written in Roman letters, it indicates we are referring about the crystal protein. When we write all the three alphabets, that is CRY, in small letters, and when it is in italics, it indicates we are talking or referring about the gene. So, there are number of types of these CRY genes present, and they are showing resistance towards or against different types of insects. Let me give you some examples. CRY1AC and CRY2AB control bollworms. CRY1AB controls corn borer. CRY3AB controls Colorado potato beetle. So, now you have understood the answer for this is CRY-IAB, which controls corn borer, that is option C. Now, let me tell you how it actually works. This gene is extracted from Bacillus thuringiensis and it is incorporated into the body of the plant or into the plant cell. This is generally done using agrobacterium. The CRY gene is inserted into the plasmid of the agrobacterium and the agrobacterium is made to attack the plant cells. Once the agrobacterium attacks the plant cell, the CRY gene is present inside the plant cell. When such a cell is made to grow into a transgenic plant, the plant will have CRY genes which starts producing the protein which the gene codes for. And once such crystals are produced or these toxins are produced, if an insect caterpillar or an insect eats it, then these crystal proteins will get solubilized and get converted into a protein. So, you may ask, if this gene is present in the bacteria, why the bacteria does not get killed? The simple reason is, in the bacteria it is present in an inactive form. So, when an organism consumes, for example an insect or a caterpillar, when it consumes the plant cell which is having this gene, the gene will produce the toxin in the form of a crystal which will not cause any damage to the plant. But once the insect eats that or consumes it, when the toxin, the protoxin or the inactive toxin, when it reaches the midgut, that is the alimentary canal, digestive system of the insect, there the pH is about 12. So, in that pH, this protoxin or the inactive toxin gets converted into the toxic form. That is, the crystals get solubilized and it becomes active. Because of this, the toxin will go and bind to the midgut of the insect. Once it binds there, it starts to bore pores over there, or many pores are formed in the midgut. This results in influx or entry of water, ions, everything into the midgut of the insect and swelling. Finally, it will burst and kill the insect or the caterpillar. This is how the toxin actually works. So, there are different types of toxins produced by different forms of this gene. Accordingly, corn borer is controlled by CRY1AB. The first two options, CRY-AC, CRY2AB, those two control bollworm in cotton plants, that is BT cotton. So, we are aware that these days there are many BT plants present like BT brinjal, BT soybean, BT tomato, all these provide resistance against one or the other insect. Statement A. Replacing the defective gene by insertion of a normal functional gene is called gene therapy. Statement B. The first clinical gene therapy was done for the treatment of AIDS. The options are both the statements A and B are correct, both the statements A and B are wrong, statement A is correct and B is wrong, statement B is correct and A is wrong. Gene therapy. Gene therapy is a technique of replacing a defective gene by a normal functional gene. This will cure the defect. This technique of replacing the damaged or the non-functional gene with a functional normal gene is called gene therapy. There are number of genetic diseases or hereditary diseases which gets passed on from one generation to other generation and which cannot be cured. For example, hemophilia, sickle cell, SCID, muscular dystrophy. These are some hereditary diseases which keeps on passing from one generation to the other generation, and there is no permanent cure for this. These defects were observed properly. These were caused because of defect in a single gene. That is, it is a monogenic defect. So, if that defective gene is replaced by a functional gene, then the defect can be cured. So, the first trial of gene therapy was done on a 4-year-old girl who was suffering from SCID, that is severe combined immunodeficiency. What is this defect? Here, the person who is suffering from SCID will have a very weak immune. It is a killer disease. A person who has this defect will have a very weak immune and will be always prone to one or the other disease and finally succumb to it or will die because of this particular disease. So, here why this disease occurs? The reason for this is SCID. It is caused because of lack of production of ADA, that is adenosine deaminase. When this enzyme is not produced, there will be no functional lymphocytes produced too. And you are aware that lymphocytes are very important to provide immunity or to provide a strong immune system to our body. So, why this particular ADA or adenosine deaminase is not produced? It is because the gene which encodes for that protein is defective over here. So, if that ADA gene is replaced by a functional gene, then it will start producing ADA and the symptoms will be cured. So, this was done for the young aged girl who was this was done for the 4-year-old girl called Ashanti De Silva. How was it first done? The first step, how it was done. So, here from her body the T lymphocytes were extracted, that is from the bone marrow T lymphocytes were extracted. Then a functional ADA gene was incorporated into a retrovirus. This virus was made to infect T lymphocyte. Once it infects the T lymphocyte, the functional ADA gene will be incorporated or introduced into the T lymphocyte. Now, this T lymphocyte which has the functional gene is again inserted into the bone marrow of the girl. Now, this functional T lymphocyte will start undergoing multiplication, and when it multiplies and when it undergoes translation and produces the enzyme, that is adenosine deaminase, then the symptoms will be cured. But this is not a foolproof kind of a treatment, that is, it has a demerit also. What is the demerit? The demerit is T lymphocytes, they have a definite lifespan. So, they are not like immortal. So, after a few days they will die. So, this treatment has to be given frequently to keep the person fit and fine or to keep them alive. So, here you can see that the statements given are replacing the defective gene by insertion of a normal function gene is called gene therapy. That is actually a right statement. The next one, the first clinical gene therapy was done for the treatment of AIDS, is a wrong statement, because the first clinical gene therapy done was for SCID, that is severe combined immunodeficiency disease. So the right answer is option C. My dear students, till now I have tried to discuss about some of the important concepts of biotechnology. While explaining these, I have given you certain information about all these concepts. So please read it properly or try to study it properly, so that if the questions are asked in some other form, you should be in a position to answer it. So wish you all a very good luck for your CET exam. This is Sushma Mani. Namaskaram.